I was just reading the latest clinical data on vascular barrier integrity in the aging brain, and it changes how we should evaluate blood-brain barrier breakdown. The brain is normally protected by a highly selective cellular wall. We often assume that age-related cognitive decline begins directly within the neurons themselves. However, the mechanistic reality is that the brain's primary vascular defense system may fail first, exposing delicate tissue to systemic stressors. By tracking the subtle breakdown of this barrier, we can identify very early markers of decline. This shifts our diagnostic focus from late-stage neural damage to early cellular vascular maintenance in the aging brain. How does this protective interface fail as the organism ages? When the vascular cells lose their tight connections, the brain is exposed to circulating components that do not belong in the central nervous system. This vascular leakage is associated with a decline in neural performance. Before we observe structural loss of synapses, the cellular barrier undergoes a slow decline in integrity that alters the local microenvironment. Here is the mechanistic question. How does cellular failure at the blood-brain barrier link vascular dysfunction to cognitive decline? We must examine the tight junctions and clearance systems that keep the brain clean. In the aging brain, we observe a gradual accumulation of inflammatory signals. This local inflammation is often a consequence of microscopic leaks in the capillary wall which allow toxic proteins to seep in. These entering proteins, such as fibrinogen, are not supposed to cross the barrier. Once inside, they activate microglia, the brain's immune cells, triggering a chronic inflammatory response that damages nearby healthy synapses. Simultaneously, the clearance of cellular debris is compromised. The brain relies on a continuous flushing mechanism to remove metabolic waste, but a compromised barrier disrupts the pressure gradients needed for this flow. Separate the mechanism from the outcome. The visible loss of memory is not the initial event, it is the downstream result of long-term cellular vascular failure and subsequent neuroinflammatory response cycles. As the capillaries become leaky, the local neural microenvironment becomes increasingly hostile. Neurons struggle to maintain their resting potential and communicate efficiently when surrounded by inflammatory signaling molecules and plasma proteins. This creates a destructive cellular loop. The chronic activation of immune cells further degrades the vascular lining, accelerating the breakdown and creating a wider field of tissue vulnerability in key brain regions. To understand this vascular breakdown, we must look at the neurovascular unit. This structure consists of endothelial cells, pericytes, and astrocytic end feet, working together to maintain the highly selective barrier. Endothelial cells line the interior of brain capillaries. They are joined by tight junctions, specialized protein complexes that seal the space between cells and prevent the unregulated passage of large molecules. Pericytes wrap around the endothelial cells, providing structural support and regulating capillary diameter. They play a critical role in maintaining the barrier during development and throughout the lifespan of the organism. Astrocytes extend cellular processes called end feet that wrap around the vasculature. These end feet act as a secondary barrier and release signaling molecules that help maintain the tight junctions between endothelial cells. Under normal conditions, this neurovascular unit restricts the entry of pathogens and large proteins. It also facilitates the transport of glucose and oxygen, keeping the brain's internal environment stable and nourished. Now, as the brain ages, this coordinate structure begins to degrade. Pericytes are lost, endothelial cells thinned, and astrocytic end feet retract from the capillary walls, weakening the overall barrier integrity. This structural degradation compromises the tight junctions. The specialized proteins that seal the gaps, like claudins and occludin, decrease in abundance, allowing parcellular leakage of systemic molecules into the brain. This leakiness is not uniform. It often begins in specific brain regions, such as the hippocampus, which is critical for memory formation and particularly vulnerable to early vascular cellular changes. The cascade accelerates when blood-derived proteins enter the parenchyma. Fibrinogen, a blood-clotting protein, is particularly damaging because it binds to microglial receptors, initiating a cascade of inflammatory signaling pathways. This microglial activation leads to the release of reactive oxygen species and inflammatory cytokines. These molecules are toxic to neurons, impairing synaptic function and contributing to cellular stress in the microenvironment. Furthermore, the presence of blood proteins inhibits cellular clearance. The brain's glymphatic system, which clears waste during sleep, relies on convective flow that is disrupted by changes in vascular permeability. As metabolic waste accumulates, it forms toxic aggregates. The combination of impaired clearance, chronic neuroinflammation, and vascular leakage creates a hostile environment that compromises the survival of nearby active synapses. Follow the evidence boundary. We are observing a clear association between vascular breakdown and cognitive decline. The cellular failure of this barrier may precede neural dysfunction by years, if not decades. As tight junctions degrade, the entry of systemic albumin also increases. Albumin uptake by astrocytes triggers the transforming growth factor beta pathway, which alters cellular gene expression and promotes inflammatory astrogliosis. This reactive astrogliosis further compromises the neurovascular unit. Astrocytes lose their capacity to support neighboring neurons, impairing potassium buffering and neurotransmitter recycling, which directly degrades the quality of synaptic transmission. The progressive loss of pericytes also leads to a localized reduction in cerebral blood flow. Neurons facing this constant energy deficit are less resilient to the toxic environment created by vascular leakage. This state of chronic hypoperfusion reduces the delivery of oxygen and glucose. The combination of low energy and high inflammation accelerates the degradation of synaptic connections, leading to network-level communication failures. So, the brain's cognitive networks must work much harder to perform basic tasks. This increased work accelerates cellular wear and tear, exacerbating mitochondrial strain and oxidative damage in surviving active neurons. The cellular debris from these damaged synapses further activates resident microglia. This forms a chronic, self-reinforcing cycle where vascular cellular failure drives neuroinflammation, and neuroinflammation drives further vascular degradation. Over time, this localized damage spreads along the vascular networks. Capillaries in adjacent regions show similar signs of degradation, expanding the area of cellular vulnerability and network dysfunction across the brain. The degradation of this vascular barrier also impairs the transport of essential nutrients. Transport proteins on endothelial cells, such as glucose transporters, decrease in density, further starving the active brain. This local metabolic starvation compromises cellular repair mechanisms. Neurons can no longer maintain their complex dendritic structures, leading to a gradual retraction of synapses and loss of functional neural connections. This final stage represents the transition from localized vascular leaks to widespread network instability. The brain's cognitive reserves are steadily depleted as the vascular cellular barrier continues to break down. To evaluate this model, we must establish its clear boundaries. Not all brain areas are equally affected by this process. Some regions maintain barrier integrity even during advanced aging. The important limitation is that direct observation of capillary leakage in living human subjects remains difficult. Most of our mechanistic understanding comes from conceptual models and laboratory tissue studies. Additionally, barrier leakage is not a guaranteed cause of cognitive decline. Many individuals show vascular breakdown under imaging without displaying any measurable symptoms of dementia or memory impairment. We must also distinguish between general aging and specific disease states. Vascular changes are normal components of aging, and they do not automatically lead to severe neurodegenerative pathology. However, in conceptual models where tight junction proteins are genetically preserved, tissues show significant resistance to neuroinflammation. This provides a theoretical case where barrier maintenance preserves local neural health. This mechanism may not explain cognitive issues driven by non-vascular pathways. It specifically describes a vascular-centered route to cognitive decline, which may coexist with other distinct biological aging processes. By defining these boundaries, we clarify our focus. We are looking at a measurable vascular vulnerability marker, not a deterministic pathway that guarantees cognitive decline for every aging individual. This brings us back to the primary question of how the aging barrier fails. The cellular wall degrades through the gradual loss of parasites and the opening of tight junctions. The brain does not fail overnight. Rather, the subtle cumulative entry of blood-derived proteins like fibrinogen acts as a hidden signal, initiating long-term neuroinflammatory pathways that damage nearby synapses. By focusing on barrier integrity, we can reframe cognitive decline as a system-level vascular failure. This cellular perspective allows us to evaluate risk before permanent neuronal loss has actually occurred. Maintaining vascular health is therefore a primary defense. When capillary blood flow is optimized, the cells forming the blood-brain barrier receive the oxygen and nutrients needed to support tight junctions. Simultaneously, supporting the lymphatic clearance system may help remove waste before it accumulates. This waste clearance is critical for preventing the inflammatory loops that degrade the vascular lining. What matters clinically is detecting these changes early. A focus on cellular clearance and vascular integrity shifts our approach from late-stage symptom management to early support of the brain's natural defenses. This perspective highlights the critical importance of vascular maintenance. By protecting the cells that line our cerebral capillaries, we support the optimal cellular environment required for healthy, long-term, complex cognitive function. So we must view the blood-brain barrier not as a static wall, but as an active, dynamic cellular interface that requires constant metabolic support to protect the neural tissue. When we support this localized metabolic maintenance, we may help preserve cognitive health span over time. Protecting the barrier is about maintaining the selective boundary that keeps the brain's local chemistry stable. How can we apply this vascular framework in practice? We can focus on low-risk behavioral protocols that support capillary circulation and promote overall vascular cell function. One primary factor is managing cardiovascular health and risk markers. Actions that support capillary integrity throughout the body also protect the delicate endothelial cells lining the brain's microvasculature. Regular physical activity is associated with improved vascular function. Consistent exercise supports blood flow and helps maintain the capillary networks that supply oxygen and glucose to brain tissue. Quality sleep is also critical for supporting lymphatic clearance. The brain's clearance pathways are highly active during deep sleep stages, helping to flush out entering waste products. Additionally, sleep support helps reduce chronic inflammatory signaling in the brain. This sleep-driven reduction in local inflammation may help preserve the integrity of tight junctions over time. Compare your current daily routine with these vascular support principles. Simple, consistent adjustments in sleep and activity patterns are low-risk ways to support your brain's natural defenses. Remember, these behavioral adjustments are not disease treatments. They represent a general framework for metabolic optimization and healthy cognitive aging, not individualized medical protocols or therapies. Track your sleep and activity consistency to reinforce these daily habits. A healthy barrier is a fundamental component of healthy aging, protecting the selective wall your nervous system needs. By maintaining this vascular cellular boundary, we support long-term cognitive health span. Optimize the protocol, protect your brain, and I'll see you in the next data set.